Hello, everybody. Welcome back. I'm here with Jordan. Jordan, how's it going?
It's going well, thanks. How are you?
Good. We've interviewed you before. It's just been a while, and this pandemic has really created a whole bunch of protocols, a whole lot of research activity in clinical research. You work for Progenibiome, as you can see here. Jordan writes most, if not all, the protocols, right?
Mm-hmm.
And we actually got a question today from somebody who contracts with major drug companies. And they were saying, you know what, how do you go from a skeleton of a protocol to the actual protocol to a study? And let's just break that down. How do you go from an idea to a skeleton of a protocol? Because you do this all the time.
Yeah.
So how do you do it? What's the Jordan way?
Well, first you have to examine your hypothesis. Make sure that what you do is going to accurately and definitively test, is that hypothesis correct? So then you come up with your hypothesis and your null hypothesis, and you figure out what tests are going to be needed in order to achieve that. Then from there, you kind of make an outline of the steps. Say I want to test a patient with autism. I would have to know, what are the standardized tests that are used to evaluate a patient with autism? How frequently are they done? Who does them? Is it a parent filled out questionnaire, or is this something that is done by a professional psychiatrist or psychologist in their office? And then from there, you determine where your intervention fits in with that framework.
Okay.
So say I'm testing a drug that's going to be helping autism symptoms. How long do I need to give this drug before I'm going to see effects?
And how do you know this stuff? Are you going on Google Scholar and looking at previous studies, or is it just from experience that you've had that you kind of know the time frames and things like that?
Well, the time frame, if it's a new drug, the time frame is going to come from animal studies.
Okay.
And possibly studies that have used drugs that are in the similar family or class.
Okay.
If it's a drug that's already on the market and we're possibly considering it for new applications, then we'd have a better idea of when it would reach the therapeutic values. But that's another thing you'd have to test.
Gotcha.
You're going to be testing your pharmacokinetics and your pharmacodynamics to see, when does this drug reach peak concentration in the bloodstream? How is it handled by the body? I mean,
Wow.
Those are things that you have to take into consideration as well.
That's amazing. A lot of people don't understand. They just understand, maybe from my audience, once you get into the clinical research part. But by the time the study actually starts, there's people like you that have been doing a lot of hours and hours of sleepless nights. Right? I mean, you've written so many protocols just in this year alone. So how do you go then from that protocol skeleton to the actual protocol? How do you go from your rough sketch or skeleton of it to the actual protocol that you're going to submit to an IRB, right?
Right. As we go through the steps, once we have the skeleton developed, then we have to kind of flesh it out, if you will. And from there, you look at each step and say, okay, what do I need to achieve this step? If I have a test that needs to be done by a psychiatrist, then this visit's going to occur in a psychiatrist's office, and they're going to do ABC. But if I need to have blood drawn by this patient, then that probably can't happen in that same visit, so we need to make a separate visit for this patient to see a nurse or a phlebotomist.
I see.
To have blood drawn. And it's just a matter of putting the pieces of the puzzle together.
So you have to have a unique skill set, because you have to not just understand from an academic perspective, what needs to be done, but you have to understand from a practical perspective. Hey, there's no way this patient's going to come here, do all these rating scales, and then have to do blood as well, right? So who's, is that you coming up with this? You and the PI, or whom are you collaborating with to come up with these schedule of assessments and exactly when everything's going to flow?
Well, it definitely is a process that takes many, many revisions as we figure out what's going to work best for patients, especially when you're talking about a group like, say, autism, where the patients aren't necessarily going to be able to do a lot of things in one visit. So yes, it's a conversation between me, the PI, and the rest of the study team. And with the
Okay.
autism example, it would probably, the conversation would also include a psychiatrist or a psychologist, somebody who specializes in autism, to say, okay, here's what we can and cannot do.
Gotcha.
And then we build the schedule around what the patients can do.
Gotcha. And then once you feel like you have a working protocol, you send it to the IRB, right?
Well, we send it to the FDA first.
FDA first, okay.
Yeah, FDA
okay.
first, and then they say, okay, we like this part of it, but we definitely need you to change this.
Ah, okay.
And sometimes they will require to see the consent documents to make sure that we are accurately conveying to the subjects and their families what is going to be happening. And there's a whole bunch of paperwork that goes with all of
Okay,
that.
and you do all this stuff? Mm-hmm. Wow, okay. Jordan, wear many hats. So
Yeah.
FDA first, how long is that process? Like from when you have the protocol you think is ready to submitting it to the FDA, how long do they usually take to turn it around?
Well, it depends on, with this pandemic going on, if it's a pandemic-related protocol, the turnaround time is very fast, much faster than you would typically expect from a government agency. But if it's not a pandemic-related protocol and we're not in the middle of a pandemic, the process takes months.
Okay, like 3, 6, either of those, 9 maybe?
Yeah, it just kind of depends on how well you put all your ducks in a row before you
Wow.
send it to them.
Yeah.
And how well you've read the regulations and complied with everything, making sure you're taking good care of your subjects, but also just doing good research
Right.
so that you're getting data of value
I
from
see.
your study.
I
Because they
see.
want to make sure that nobody's wasting their time, that we're doing something that's going to bring science forward.
And your official title is medical writer? Is that okay? Wow. And this, how did you get into that career?
I have a bachelor's in biology from McMurray College in Jacksonville, Illinois, and ironically, I always wanted to be a veterinarian.
Okay.
So I have a lot of experience in the animal world, working as a registered vet tech, which exposed me to a lot of medical terminology that I wouldn't otherwise have known.
Gotcha.
And then I have a master's in biotechnology from California State University, Channel Islands, where I learned a lot of the nitty-gritty of the science of things, how the regulatory process works, the different phases of drug development, and putting those hats together kind of makes a non-traditional medical writer.
And you've even had a clinical research coordinator experience, which is invaluable now for your future writing. How much has your experience as a coordinator affected how you write protocols? Do you think it's made you better at understanding the process?
Absolutely. It's immensely impacted the way I write protocols because having seen how a protocol is implemented from the ground position, oh my gosh. I have so much sympathy for clinical research coordinators.
Yeah.
They have to take these ginormous documents and translate them and make sure that they are doing everything accurately, that they're collecting good data. And
Yeah. Wow.
the better written a protocol is, the better it enables them to do their jobs well.
You're definitely probably one of a kind, like maybe just a handful, like a couple dozen people like you in the country, maybe in the world, that have that kind of experience. Seriously. So hats off to you guys here and at Progena Biome for doing that. And thank you very much for explaining the, I guess, do you call that preclinical process? Or it's not really
Preclinical is like
right?
animal studies.
Okay. So what would this be? Like pre-research trial, I guess, protocol development phase?
Yeah,
I don't
something
know. It
like
doesn't
that.
sound great, but thank you very much for explaining this to us because, like I said, most people watching, they just understand from when the study starts. They don't necessarily understand all the back end of how protocols get created and things like that. So thank you very much, Jordan. I appreciate it.
My pleasure.